So this is our second lecture, I think, isn't it? So we had the introductory part in the previous class. So there we have seen the broad overview of the syllabus and the topics associated with this portion of general studies. Now, picking up the thread from the previous class, from the last topic that we ended. So what was the last topic that we ended in? Composition of blood cells. Okay. This part is done. Okay. So last lecture we discussed on composition plus what is the normal concentration of red blood cells, isn't it? White blood cell and the platelets. Now let's see more on these individual blood cells, about their functions. Now coming first to the red blood cell. So last class we discussed that the scientific name of red blood cell, that is the erythrocyte. Difference between antibiotics and antibacterial. See, antibacterial medicines are called as antibiotics. Then gut microbiota. It will once again come in our discussion when we are going to discuss on bacteriotherapy. That beneficial microbes present in the intestine, including small, large intestine, come under the gut microbiota. So different microbes that have colonized inside our body, in our gut, that become part of the gut microbiota. These are commensal microbes that provide benefits to our body in boosting the immunity. Coming to the erythrocyte, we can see here erythrocyte or red blood cell. So this appears a disc-like shape, In the middle we can see two depression in the middle, that gives the name biconcave. Biconcave nature of RBC, this increases flexibility in the RBC. Flexibility means that it can change its shape, just like we all know about amoeba, can easily take any shape, isn't it? It can squeeze itself, that is, it can shorten in size, reduce in its size. Similarly, our red blood cell also has that flexibility, that they can change their shape. They can squeeze themselves to narrow themselves or shrink in their size. Why this is needed? Because now we know that blood flows inside the blood vessels, isn't it? Blood flows inside what? Blood vessels. Blood vessels like arteries, vein. Artery, we say that it carries oxygen-rich blood. Arteries carry oxygen-rich blood, or simply oxygenated blood. Veins, they carry deoxygenated blood or carbon dioxide rich blood. That is the functional difference in artery and the vein. What is gastrointestinal tract? This includes mouth, esophagus or food pipes, stomach, small intestine, large intestine. These are all part of GIT, also called gut. ACE2 we discussed in the name of ACE2 receptor that is required for maintaining the blood pressure. But they stick the coronavirus also, unfortunately. So blood vessels, so blood flows inside these tiny tubes called blood vessels. Now these blood vessels are not of uniform size in our body. Suppose blood vessel which is carrying oxygen-rich blood to the kidney, to the liver, they may have to branch into smaller arteries. And the smallest blood vessel we call it as the capillary. So this is single cell thick or thin, single cell thick or thin. So this is the thinnest, you can say, blood vessel, that is called capillary. So these are branches of arteries and the veins. So if they have to enter into a space like your kidney, your liver, they branch into smaller arteries, smaller capillaries further. Now, smaller means here the diameter of this capillary will be narrower than the diameter of the larger blood vessels. So diameter will be narrower than the arteries and the veins. These are the branches only of them. So when RBC is flowing inside the blood vessel, suppose this is the larger blood vessel, and now what it does, it is showing that it is branching, isn't it? So diameter is getting narrowed. So when the diameter narrows down, and this is your RBC. Suppose we say that these are capillary. Now capillary diameter is greater than the diameter of our red blood cell. Here we are showing that this is the blood vessel. This is the blood vessel. Now this blood vessel diameter is larger than the RBC. RBC is able to make its way forward. So this is the blood vessel where we are showing that is the blood flow. We are showing the blood flow. Blood flow means it involves RBC also flowing along with the blood. So here we are showing that this blood vessel diameter is larger than the RBC diameter. But as it rebranches, then into tiny capillary, so capillary diameter will be narrower than the diameter of RBC. So how this RBC will make its way forward? So now it has to squeeze itself. It has to narrow its size. So same RBC now. So this is the larger diameter it shows. So it has to squeeze itself to get accommodated in the tiny blood vessel. So before it enters here, it has to squeeze itself. So that flexibility will come due to biconcave nature of RBC. So RBC we have written what is biconcave. And this biconcave nature of RBC gives flexibility so that it can accommodate in tiny blood vessels including capillaries. Why it is important to know? Because when we are going to discuss a genetic blood disorder that all of you know at least the name sickle cell anemia. In sickle cell anemia, RBC loses that flexibility, it becomes rigid in nature and it will block the blood flow. So in people with genetic blood disorder, sickle cell anemia, here the RBC becomes curved and elongated in shape. RBC becomes what? Curved and elongated. So instead of this disc-like biconcave shape, RBC takes this crescent moon shape. So this is the RBC. and hemoglobin inside the RBC. We are ready to discuss what is sickle cell anemia. Sickle cell anemia is because of defective hemoglobin. Because of what? Defective shape of hemoglobin. So basically it is a hemoglobin disorder. Hemoglobin disorder. Hemoglobin disorder is such that the shape of RBC here becomes what? Curved and elongated or sickle shaped. Because of this reason, now the RBC becomes rigid in nature. This RBC is what? Rigid. It loses its flexibility. In sickle cell anemia, genetic blood disorder, this is because of what? Defective hemoglobin, where the shape of hemoglobin changes and it changes the shape of RBC to become what? Curved and elongated or sickle shaped instead of biconcave shape or disc-like shape. So that is how, what will happen now in these individuals with sickle cell anemia. Suppose this is the blood vessel and these sickle shaped RBCs they are making their way forward, isn't it? Now, when this blood vessel branches into tiny capillaries, just now we discussed that, suppose it branches into what? Tiny capillaries, which we discussed single cell thick or thin, single cell thick or thin. So its diameter is narrower than what? The RBC diameter, just now we discussed. But as there is rigidity, so there is what? So these are rigid sickle shape RBC, which do not have flexibility. So what will happen now? Can you tell me? So in tiny blood vessels, capillaries, what will happen? This is going to block the blood flow. Suppose this blood is moving towards kidney, blood is moving towards brain or heart, what will happen? It blocks the blood flow. In tiny capillaries. So it will clog, isn't it? Just like the pipe that carries water, isn't it? If it's too narrow, what will happen? It will make sluggish now the movement of the blood. And what is the role of RBC? RBC role is to carry oxygen. How oxygen will reach now to the body organ? So these sickle shape RBC, they aggregate. What will happen? They are aggregating, isn't it? Aggregate and block the blood flow further. And gradually these are destroyed also in large amount, causing severe anemia. So that is why it becomes fatal condition, life-threatening condition, sickle cell anemia. We have just discussed symptom, what happens sickle cell anemia. The background reason is genetic reason, which we are yet to discuss. Is it clear now to all of you that in sickle cell anemia, because of defective shape of hemoglobin, it changes the shape of RBC to become curved and elongated, and it blocks the blood flow in tiny blood capillaries. And that further results in excessive destruction of this RBC hemoglobin, causing severe anemia. Now, what do you mean by the term anemia? Let's come back to our RBC discussion. See, if you see here, we have drawn the RBC erythrocyte. We are saying that it is biconcave. We understood it is give flexibility. Now, what about non-nucleated? Non-nucleated means it is anuclear. It does not have any nucleus. So it is anuclear. Anuclear means non-nucleated, does not have nucleus. Anuclear means here it is non-nucleated. Now, what is the reason it is non-nucleated? See, RBC, this is carrying the oxygen to different body cells, which is essential for our survival. We discussed that is oxygen will be needed to break down glucose into energy. So RBC has to just act as a ferry to transport this oxygen. While transportation oxygen depending upon requirement of the body. So what is the requirement of body? When you compare amoeba with that of human body, don't you think that requirement of oxygen is much more higher in human body, isn't it? So multicellular higher organisms demand of oxygen will be higher. Particularly within higher organism also mammals including human being, their demand for oxygen is much more higher than that of non-mammals. So body requirement oxygen will be higher to derive the energy. So as a feature of evolution, we can say that thousands and thousands years of evolution, mammals have evolved to meet the demand of that higher energy, to meet the demand of higher oxygen. What they have done, they have eliminated the nucleus. The reason is eliminating nucleus from RBC will provide space to accommodate this hemoglobin. So hemoglobin is present where inside the RBC. It has to provide space to accommodate hemoglobin. Presence of nucleus will reduce that space. Presence of nucleus will reduce the space or surface area to accommodate the hemoglobin. So that is the prime reason why there is no nucleus in the RBC. It increases the space to accommodate the hemoglobin. Now what is hemoglobin? Hemoglobin, chemically it is what? A protein. Protein with what? Iron. The chemical composition of hemoglobin is, it is protein plus iron, hemoglobin. This hemoglobin, from where it comes in our body? It is not that you are consuming some food and that food contains hemoglobin. Our own body will manufacture, isn't it? Using the raw materials or proteins to make the final hemoglobin. So we have to provide to the body iron and protein source, isn't it? From the food source. And this will be finally converted, transformed into the final hemoglobin. Our own body has to process and produce the hemoglobin. Inside the red blood cell, it gets accommodated to carry oxygen. So hemoglobin chemically is a protein with iron. As it carries oxygen, so oxygen combines with the hemoglobin. Oxyhemoglobin that transports oxygen to different body cells. So till now we discussed that RBC transports oxygen. How does it transport oxygen? It is in combination with the hemoglobin inside the RBC. If there is lower concentration of hemoglobin, it will reduce ability of RBC, ability of hemoglobin to carry oxygen. Now people with anemia, people with what? Anemia. Anemia means there is deficiency of, yes, so deficiency of RBC, deficiency of iron, deficiency hemoglobin, all these can result in what? Anemia. Any one of them, any two, all the three, isn't it? So any one, any two, or all the three can result in anemia. Now, if a person has deficiency of iron in the body, this iron is coming from food source. If the food source that we are consuming, our diet is deficient in iron in day-to-day life, then automatically that will reduce the production of hemoglobin in your body. And that is going to affect the oxygen carrying capacity also. So which function of the body will immediately get affected because of anemia? Transportation of oxygen, isn't it? In anemia, the first vital function which is needed for our survival, transportation oxygen, gets affected now. And without oxygen, how the bodies are going to produce energy to continue their life processes? It will be difficult. So in case of anemia, we have written any of the reasons could occur, but you can write first of all normal hemoglobin concentration. What is the normal hemoglobin concentration in a healthy individual? So healthy person should have, yes, how much hemoglobin should be there in your body? 10 to 15. 12. I will tell 14 to 15 gram. This is the normal hemoglobin concentration, 14 to 15 gram per 100 ml of blood. It will vary. Definitely in males it will be higher and lower in case of females. In males 15 gram, in females 14 gram. So this is the ideal value for hemoglobin in your body. In males 15 gram per 100 ml of blood, optimum level. In females 14 gram. Now any value that is less than will become severe anemia. So what is that value? 8 gram per 100 ml. Any value that is less than 8 gram, that becomes severe anemia. To lead a healthy life, you also need some basic data, isn't it? The most important test immediately a person has to go for what? Blood test, the CBC test. So under that, your hemoglobin concentration is vital. Because if you have this form of anemia where the hemoglobin level has come down to less than 8, then immediately your oxygen carrying capacity will get affected in the body. So this reduces oxygen supply to your body immediately, significantly. So first of all, person with anemia, what could be the symptoms here? Lack of energy means what? Here it will result in weakness, fatigue, headache. So even doing some basic activities also, person feel fatigue. The tiredness is now getting exhausted in terms of fatigue. Then person can even faint also, isn't it? 20% of glucose and oxygen is required by the brain. Overall oxygen requirement 20% required by your brain continuously. So there is also risk of what? Anywhere this person can faint anytime. It will be dangerous. Immediately person has to go for blood transfusion, isn't it? Because it will take time to take the food source and then increase the hemoglobin content. Now this anemia could be due to a number of reasons. It could be because of dietary deficiency or some medical condition that also can cause anemia. Why this is important for us to know is because in India 50% females, they are said to be anemic, 50%. So this is widely prevalent, not only in females but also in children. So children, women, whether it is pregnant females, lactating females or females of reproductive age, everywhere we have found in India that the 50% they are anemic. So that is why it is a concern for us. It is said that anemia in mother is more dangerous than anemia in the child. What is the reason? Anemia is more dangerous in mother than the child. Transferred genetically. Now, As I told you that anemia could be due to number of reasons: dietary deficiency, medical conditions, any one of them. So if you look for reasons for anemia, then this could be nutritional deficiency, isn't it? This can be due to what? Nutritional deficiency, or this can also be due to some medical conditions. Just now we have discussed sickle cell anemia, isn't it? So this is a medical condition, but this is a genetic blood disorder. So there is excessive destruction. In sickle cell anemia, there is excessive destruction of hemoglobin and RBC. Lifespan, here it is just 10 days. Lifespan required is 120 days. But here in 10 days only destruction of RBC hemoglobin occurs prematurely. So this is an example of what? A medical condition. It's a genetic blood disorder. So basically it is hemoglobin disorder, isn't it? The genetic blood disorder is related to what? Hemoglobin disorder. Or hemoglobinopathy. Hemoglobinopathy means the same medical condition of hemoglobin disorder. Now, anemia can also be due to cancer, isn't it? So, people undergoing chemotherapy. So chemotherapy means taking anti-cancer drug molecules. So these anti-cancer drug molecule, when cancer patient they have to take, its role is what? Its role is to destroy the cancer cells, isn't it? However, when this anti-cancer drug molecule, suppose this is the cancer cell means these are showing uncontrolled cell division. They are showing what? Uncontrolled cell division. So these anti-cancer drug molecules, they enter in your blood and then absorbed by, so these are fast dividing cells which will absorb from your blood anti-cancer drug molecule. This anti-cancer drug molecule will kill the cancer cells. However, these anti-cancer drug molecules, they do not know which is a cancer cell and which is a healthy cell. So they will also be absorbed by healthy dividing cells, healthy dividing cells also. Or you can simply write healthy cells. So anti-cancer drug molecule, it also enters the healthy cell. So unfortunately, it also kills the healthy cells. For example, blood stem cell present in bone marrow. Blood stem cell present in what? Bone marrow. We'll discuss that what is this bone marrow, blood stem cell. So unfortunately, it also suppresses these healthy cells. So that is the side effect person has to undergo, that it will suppress, destroy the healthy cells also. Now what will happen? If these blood stem cells, from the blood stem cell, new RBC are getting produced. New RBC are getting produced from here. So by suppressing it, it will cause reduction in what? The normal RBC, which are produced in bone marrow, then they come to blood circulation. So automatically this will cause what? Anemia, chemotherapy induced anemia, which is inevitable. So there is a benefit of treatment, there is a risk of treatment. When we see that benefits of treatment far outweighs the risk of treatment, we adopt that treatment, isn't it? In the larger interest to protect the person's life, because once we withdraw this chemotherapy, once again the blood stem cell will start producing the new blood cell. It can restore. But initially we have to boost by various means, through food, through some drugs that can induce increased production of these blood cells in those body. But this also can result in what? Anemia. Now anemia, medical condition, can also be due to some parasitic infections. Parasitic anemia. This is common in children, isn't it? School going children, toddlers, they have a tendency to put their what? Every time their fingers into mouth unknowingly, less than 5 years old. So they may get infected with what? Some parasites. So that also can result in anemia. One common form example you can write also even the malaria also can cause what? Anemia as a parasitic infection. Although it is transmitted by mosquito acting as a vector. So malaria is vector-borne disease. Vector-borne disease, where vectors are female Anopheles, mosquitoes. And the causative agent parasitic protozoa, Plasmodium. So this also can result in anemia. How it results in anemia? This Plasmodium, when enters your body, so person X already infected with malaria, which means in the person's blood there is presence of Plasmodium P. So malaria is caused by what? Parasitic protozoa Plasmodium. It is transmitted by the mosquitoes, female Anopheles mosquitoes. So person X already infected with malaria means there is presence of Plasmodium P in the blood of person. Now when mosquito M bites person X while sucking the blood, Plasmodium P enters the body of mosquito M. Now this mosquito M carrying the Plasmodium P, when bites healthy person Y, will transmit Plasmodium P to healthy individual Y. So mosquito M acting as what? Vector or carrier to carry the infection. So person X already infected with malaria, having Plasmodium P in the blood, bitten by the mosquito M. While sucking the blood, the mosquito will also get in what? The Plasmodium P. Now this mosquito M carrying the Plasmodium P of malaria bites healthy person Y, it will transmit the infection. So mosquito acting as a vector, as a carrier to transmit the malaria infection. This just first of all, we are trying to discuss on anemia. These topics will come later on also. But how do we associate with anemia? So anemia can be due to some medical conditions, cancer, sickle cell anemia, genetic anemia, or it can be due to some parasitic infections also. Now regarding nutritional deficiency, protein malnutrition, malnutrition can cause anemia, because ultimately for the body to produce hemoglobin, protein, it requires the raw material of protein food source. If there is deficiency protein in your body, then automatically body will not be able to provide raw materials or amino acids to make the final protein. So building block of protein is what? Amino acid you get from the food source. Then if there is deficiency of iron in your body, because even if protein is there but you have iron deficiency, still your body cannot make the final hemoglobin. Then another one is vitamin deficiencies. Vitamin deficiency. Under vitamin deficiency, some important ones are folic acid deficiency. Folic acid means vitamin B9. So one of the B complex vitamins, vitamin B9, is folic acid. Why we have taken this? Because one of the prominent reasons for anemia in India. So what is the reason for, we discussed 50% females anemic? Can you tell me? What is the reason for anemia in India on large scale? Iron deficiency anemia, followed by folic acid deficiency. But main answer is what? Iron deficiency. So nutritional deficiency of iron is a major cause of anemia in India in females. This is followed by folic acid deficiency. These are the two major reasons. But prominent reason is iron deficiency. So we need to connect to the society from the topic. So now we are able to connect now that in India, if 50% females are that is anemic because of what? Iron deficiency, followed by what? Folic acid deficiency. So government should intervene, should try to resolve this issue with the target of what? Making India anemia mukt Bharat Abhiyan. So we have a Poshan Abhiyan, Poshan Abhiyan Yojana, yes. Why? Why in females you are asking that? Now see, if you consider, just now we discussed that hemoglobin in females slightly lower than what? The males. The reason is biological demand for iron, vitamins will be higher, because biological loss of blood is higher in case of what? Females, for natural reasons. Isn't it? So menstrual bleeding is there, that will reduce automatically what? The composition of the blood in terms of the hemoglobin and also the iron. So these are biological reasons. So therefore biological demand of iron, biological demand for these protein will be higher in case of what? The females. Then for pregnant females it is in addition, isn't it? In pregnant female there is a fetal blood and there is a maternal blood. So to form fetal blood, definitely there is excessive requirement of what? In terms of than the normal requirement in the pregnant females. In pregnant females, requirement of iron will be higher and also the vitamins to support the fetus developing in uterus of mother. So considering that aspect, before we go to that, so nutritional deficiency, protein malnutrition, iron deficiency, vitamin deficiency, folic acid deficiency. There are also other vitamins like vitamin B12 deficiency, and B12 you can write scientific name is cyanocobalamin deficiency. So this is vitamin B12. Now both folic acid and B12, these are essential for the production of new RBC in your body. So just now we discussed there is production of red blood cells in your body. For production of these red blood cells, your body requires these two important vitamins. B9 also, B12 also. Let's discuss on folic acid. Because after iron deficiency, the next issue is what? Folic acid deficiency. Yes. That's what I am now going to discuss, that why it is more dangerous in mother than that of the child. So why anemia more dangerous in mother than that of the child? Folic acid, vitamin B9, essential for DNA synthesis and cell division. Essential for production and maturation of RBC. So one of the B complex vitamins known as folic acid, vitamin B9, is essential for our DNA synthesis, cell division. This function promotes life. It promotes what? Life. How do we say it promotes life? Because it enables development of vital organs, brain, heart, in fetus. So the fetus developing in uterus of mother during the first 3 months of pregnancy, what is happening? There is rapid cell division, DNA production, then only the heart, lungs and other body organ they are going to be formed in the body. And that will require what? DNA synthesis, cell division supported by folic acid. Is it clear? So first 3 months of pregnancy in mother, for the development of vital organs of the fetus, whether it is brain, spinal cord, heart, all will require rapid cell division and DNA production. And this is enabled by what? Folic acid. So that is the reason why, suppose mother has deficiency, pregnant mother has what? Deficiency of folic acid. What will happen? Abnormal development of brain, hole in the heart, isn't it? Abnormal development of brain, heart, or hole in the heart. In that case, child may not survive. Either there could be death of the fetus in the uterus of mother, or it could be stillbirth, child born was already dead, or even if survives, it will permanently damage the brain of the child, cognitive impairment, ability of the child of thinking, reasoning will be reduced compared to the normal individuals, will face deprivation in the society for no reason. So that causes permanent damage. Even if a child is born, isn't it? It causes permanent damage to the brain of the child, and it cannot be reverted back. Irreversible damage to the brain. So that is why we are saying that, so even by giving best of the nutrition to the child born to the anemic mother, we will not be able to make the child to lead a normal life. So that is why anemia in mother is more dangerous than what? Child. Every 6th person born in the world is an Indian. It is not an issue of only India, national issue. It is an international issue. So we have to bridge this gap, isn't it? So this is how deficiency of folic acid known as megaloblastic anemia. Deficiency folic acid is known as, yes. Because it is affecting the future generation. Is it clear? Intergenerational impact of this form of anemia. The future generation of the country is getting affected. Therefore, we are saying it is more dangerous in mother compared to what? The child. That is the context. So deficiency folic acid, you can write, is called what? Megaloblastic anemia. And what are the symptoms? Maturation failure, of RBC, and severe anemia. Then we can also write abnormal development of vital organs of the fetus, including brain, resulting in congenital malformation or birth defects. So it can result in what? Birth defects in the child. Birth defect here means congenital malformation. And cognitive impairment. Cognitive impairment. Cognitive impairment means here ability to think, reason will be affected permanently. So this is on the anemia, including megaloblastic anemia. Now, how to address this issue? So in the year 2018, government launched Anemia Mukt Bharat Abhiyan. So these include intervention strategies to deal with the anemia in India. So we discuss what is anemia, isn't it? And we try to also focus on some symptom of anemia, reasons for anemia. But ultimately, what is the solution to the prevalent anemia in India? So in 2018, we discussed government launched this Anemia Mukt Bharat Abhiyan. So what are the interventions that are required to deal with the anemia in India? First one, now remember, it has to be preventive measure, isn't it? We have to take preventive measure that will produce the result. So preventive means prophylactic measure. Preventive means what? Prophylactic measure is called preventive measure. So prophylactic iron and folic acid supplements in the form of tablets, distributed to females and children. So females including females of reproductive age, pregnant females, and also lactating females. So there should be prophylactic distribution from the government health centers to the females and children to fight against this form of anemia. because this is basically nutritional form of anemia that we are noticing. So how we can provide additional supplement if it cannot be through the food source. So females including reproductive age, pregnant females, lactating female means, that is those who are given birth to the young or breastfeeding females, that is the lactating females. Then another thing, food fortification. With iron and folic acid. So staple food, suppose it is rice. Staple food means what? The food that is consumed for longer duration in the population. That is a staple food. The primary energy source. For example, in some population it will be rice, somewhere it could be wheat, isn't it? So whatever the staple food, so during the processing of that food, we can mix the paste of iron. We can process with what? Powder of iron, for example. So that will help in what? Food fortification. And through that we can reach the population by targeting the staple food in the population. That is called food fortification. So during processing of food, you are adding iron paste or iron powder so that it can mix with those food items and then it can reach the population. That is called food fortification. So this is also an important strategy to reach the population. You can fight the calorie deficiency and also can go for the iron deficiency also along with that. So food fortification with iron and folic acid. This is another intervention. So wherever it is possible. Now apart from this, deworming. Now deworming means then children may or may not be infested with parasites. So parasites in their intestine can suck the blood continuously. So what we can go for, we can give them deworming drugs. To children. The most common example all of you should know is albendazole, deworming drug. To prevent parasitic anemia. To prevent the parasitic anemia. So some parasites like tapeworm may be there in the intestine, which are tearing the intestine layer and sucking the blood in the children. So that can be get rid of using these deworming drugs. Calcium supplements to prevent fluorosis induced anemia. So can you tell me what is this calcium supplement, what has to do with the fluorosis? Yes. Yes, fluorides in water, you are correct, but how it is associated with this intervention and why? So there are certain states where you may find more fluorides in their water, groundwater. That may be geological reasons. The parent rock material is having the minerals also, isn't it? So they may contain higher fluorides. So during weathering of rocks, the water they carry, so groundwater may have higher fluorides. And higher fluorides in drinking water, it can damage the intestine, The iron that we take through food source get absorbed through intestine. If it is damaging the intestinal layer, absorption of iron gets reduced. So automatically it will cause what? Anemia. So what is the solution? Have you understood the problem first? So what we are saying that excessive fluorides in drinking water damages intestinal wall or the layer. So excessive fluorides in drinking water damages intestinal wall or layer, reducing or preventing absorption of iron. Is it clear? So this is the reason. Now what will happen that calcium supplements, calcium will bind with the fluorine in water, that is the forming the fluorides, isn't it? Calcium will bind with the fluorine, forming calcium fluoride, and fluorine will not be available to damage the intestinal wall. So you can write calcium binds forming calcium fluoride removing fluorine from the drinking water. So calcium binds, forming calcium fluoride, removing fluorine from the drinking water. So fluorine will not be available in free form to damage the intestinal wall, and this indirectly will prevent fluorine induced anemia. Is it clear to all of you? Now what we are discussing in this Anemia Mukt Bharat, there are certain localized solutions also. So anemia, one is that is prevalent because of what? Iron deficiency, but there are certain localized reasons, endemic reasons, which are found there only because of that geographical area. So this is endemic anemia, fluorides in drinking water. So in order to address that endemic anemia, this is the localized intervention strategy, which is also part of the Anemia Mukt Bharat Abhiyan. So how you suit that strategy to the localized conditions of that region, so that it is able to resolve the issue. So this is another strategy, part of the same Anemia Mukt Bharat. So these are some of the important strategies as a part of what? Anemia Mukt Bharat interventions. Prophylactic iron and folic acid supplementation to women and children, food fortification with iron, folic acid, deworming drugs, calcium supplements to prevent fluorosis induced anemia. So what you have to understand is questions can be asked from the concept. This is the applied part of the concept. What is the issue in the society? How that issue can be resolved? So questions could be from government programs and policies also. That what are the interventions as a part of Anemia Mukt Bharat Abhiyan? You have to select which are the appropriate interventions as a part of this program. Or question could be directly from the concept also in the prelims exam. So whenever a topic we cover in the class or you read in the newspaper, you have to connect that how that topic is connected to the society, how it is impacting the society. Is there any issue that emerges? Then how the society is able to resolve it. Then what are the other government programs and policies? That also need to focus. That is why I told you in the first lecture to go through what? India Yearbook, where you will get the government programs and policies. Yes. So target year they have set, but it is too far in terms of that is 5 to 6 years they have set a target to address this issue. But again, we have to see that when after the general election results, how that is being, but currently we are missing the target. Yes. It could directly, just like iron tablets, calcium tablets can be given. The fluoride present in the water, is it clear? The fluorine, calcium will bind with that fluorine. Then fluorine will not be available to damage the intestine. You have to remove fluorine from the water. Therefore, calcium is binding with the fluorine. Yes. Yes, the time of clamping, as I told you, that there are a number of reasons for anemia. Yes. Yes, cord clamping also, because immediately if it is done, so then it is not able to address the issue. About 3 minutes, it says that you have to wait for clamping. So that is a part of prelims. So that is the objective question. You don't have to write the means. Is it clear? Magnesium also can do. Calcium will provide strength to the bones also, isn't it? It makes more sense. We can give magnesium also as a supplement, but that will be more appropriate giving for the calcium. Okay, now we try to discuss on red blood cell in today's discussion, isn't it? That is what is RBC, what is hemoglobin. So we have written it is non-nucleated, the red blood cell. You can also write that there is absence of mitochondria also in erythrocyte, absence of nucleus, absence of mitochondria also. So all of you know this is the powerhouse of the cell, but that is also absent in your RBC. Absence of mitochondria also. So again the reason is same, to provide space to accumulate hemoglobin. So reason is same, why it is non-nucleated, same as why there is absence of mitochondria. Mitochondria, this is the site of aerobic respiration. So mitochondria at the site of aerobic respiration. Now, can we use RBC for DNA fingerprinting? Can we use RBC for DNA fingerprinting? No, because there is absence of nucleus. There is no DNA in the RBC. You cannot use RBC for DNA fingerprinting. Is it clear? Now, if there is absence of mitochondria, then how does RBC get the energy source? What is the answer? Every cell must have what? Mitochondria, because it will use that oxygen and glucose to convert into energy, called aerobic respiration. Without respiration, how this RBC gets energy? Yes, what is the answer? All of you know the answer. We discussed that if there is no oxygen, there could be what? Anaerobic respiration. So RBC performs anaerobic respiration. So RBC for its survival performs what? RBC undergoes anaerobic respiration. So cellular respiration, aerobic, anaerobic, here it is anaerobic. Because if RBC itself will consume oxygen, then what it will deliver to the body cells, isn't it? So that is an adaptation of our body, that it should not have those cell organelles, like absence of nucleus, absence of mitochondria, accumulate hemoglobin and carry the required amount of oxygen to the different body cells to meet the demand of oxygen in the body. Yes. In the cytoplasm of the cell, through glycolysis, they are going to produce energy, anaerobic respiration. That electron transport chain will not be needed by the RBC. Okay, let's now come to the site of synthesis of blood cells or So now we can see the site of synthesis of blood cells or site of hematopoiesis. Site of hematopoiesis. So from where these blood cells are produced in your body, so continuously throughout our life, there is recycling of blood cells. Last lecture you have written 120 days for RBC, 10 days for blood platelets, few days for WBC. So continuously throughout your life there is production of what? New blood cells. So in what context we are using the term for stem cell here? Stem cell, what is a stem cell? And what is this blood stem cell? Stem cell, it comes from the word stemness, which means to originate. So the cell from which all the new cells originate, that becomes the stem cell. For new cells to originate, cell has to multiply, isn't it? There should be cell division. So whenever we are writing stem cell, we discuss that derived from the word stemness, and stemness means to originate. So the cell from which all the new cells originate in your body, that cell becomes what? The stem cell. The cell from which all the new cells originate in your body, that cell becomes what? The stem cell. And this will happen only when there are two fundamental properties associated stem cell. One is self-renewal. Another one is differentiation. So two fundamental properties of stem cells are self-renewal and differentiation. Now for self-renewal, there should be continuous cell division in this cell. Then it will form new cells. It is replenishing new cells in your body. For this, there must be continuous cell division. So self-renewal means it can replenish, renew itself through continuous cell division. So whenever there is some damage or some blood loss, new blood cells can be produced. In natural course also they get recycled. When you are donating blood to someone in blood transfusion, again the body will reproduce, otherwise it gets wasted. So throughout our life, how these new blood cells are produced? Because we have blood stem cell. Now differentiation means basically specialization. Cells are specialized to perform a specific task. Specialized to perform specific task. Or differentiate into different cell types. Differentiate into what? Different cell types. For example, stem cell liver stem cell, blood stem cell, intestinal stem cell, here are some examples. So they first divide, then differentiate into specific cell type of your body. So continuously throughout your life, new blood cells are produced, isn't it? New blood cells are produced, new liver cells are produced. So liver is the youngest, it remains ever youngest. Even if you become 90 years, liver still has the property of what? Multiplication. It will remain young in your body, because it contains abundant source of liver stem cell. Even if between donor and recipient 50% of liver donated, 50%, still it can regrow. Still it can what? Regrow. So that is the ability with the liver. That is the best example of stem cell in your body, liver stem cell. So wherever these stem cells are present in your body, that part of the body will show continuously regeneration and repair. Regeneration and repair. Wherever these stem cells are not there, that part of the body is damaged forever. There is no repair. For example, heart muscle death, that is called heart attack. That is forever. That part of the heart muscle cell is damaged forever. Spinal cord injury is forever. It cannot regenerate. So wherever your body cells can regenerate, that part of the body can show renew, repair. Wherever there are no stem cells present there, it cannot undergo regeneration. It cannot repair itself. So these are the best examples of stem cell underlying your body. There are also other examples. Skin, whenever some damage is there in the skin, isn't it? Some abrasion, new skin, you can see the formation. Because the dermis, the innermost part of the skin, that will produce the new skin cells. So two fundamental properties of stem cell, one is self-renewal. They undergo continuous cell division. Differentiation. So differentiation means they differentiate into different cell types of your body. Differentiate into what? Different cell types of the body. And they specialize to perform a specific task for the body. So liver stem cell, that will undergo a differentiation towards liver cell. Blood stem cell for the blood cell. They are performing different function. So tissue specific cells are produced by these further division and differentiation. But these are the two fundamental properties. Whenever you write on stem cell, these two properties must be mentioned. Coming back to the blood stem cell. So blood stem cells are present where? Inside the bone marrow. What is bone marrow? This is actually the hollow interior of your bone. Hollow interior of your bone. You can write it is a soft, spongy tissue inside of bones. Soft, spongy tissue inside of bones. Bone marrow. This bone marrow contains what? Inside them, a specialized area called red bone marrow. And this red bone marrow contains blood stem cells. Red bone marrow has what? Blood stem cell. Also called hematopoietic stem cell. Some bone example we have taken, like your hip bone or pelvis. Hip bone or pelvis contains red bone marrow inside which there is presence of blood stem cell. So from these blood stem cells, continuously throughout our life, new blood cells are produced, including RBC, white blood cell in the blood, platelets. All your blood cells, the site of production is what? Bone marrow. All the blood cells are produced from where? Bone marrow. That is the correct answer. Now another important location, we can see thigh bone. Now this is an example of what? A long bone of your body. The shaft of the bone. So this is called as what? Elongated part is called shaft of the bone. But red bone marrow is present at the ends of the long bones. Not in the shaft. It is in the ends of long bones. At the ends of long bones we have presence of what? Red bone marrow and blood stem cell. Not in the shaft, not in the elongated part of the bone. So this is another example of an important site for presence of red bone marrow and the blood stem cells. Yes. See, some bone, it is not that in these only we are saying, it is also present in your vertebra. It is also present in your ribs, present in your sternum. Is it clear? These are other bones. I am not saying that these are only two. These are the other examples. Vertebra, that forms your what? Backbone. Your chest is surrounded by these, made by these ribs only, and they are joining together at the level of sternum, breastbone. Isn't it? Sternum. So your ribs, ultimately they join what? The sternum. So that also contains the bone marrow. So this is important, the hematopoiesis part, isn't it? From where these blood cells are produced in your body. So some person who may have, suppose, blood cancer, bone marrow cancer, you must have heard about bone marrow cancer. What will happen? Then in that case person may have anemia, in this case also, in bone marrow cancer. We can blood stem cell transplanted into the recipient. See, immediately after birth, you do not have those well-developed bones. Is it clear? So, starting from yolk sac, starting from what? Yolk sac, when the fetus in the uterus of mother, followed by the spleen, followed by the liver, and finally going to the bone marrow. So, the correct answer is what? Bone marrow. Yes, you have to write bone marrow as the correct site of production of blood cells. So after birth, the bone marrow takes over, producing the new cells. Okay, so this is about the stem cell. This will be further discussed with you in biotechnology class in more detail. Is it clear? We have just introduced blood stem cell. It was required in our discussion. This is a separate topic to be dealt under the topic biotechnology. Like what is stem cell therapy? What is regenerative medicine? What are the Nobel Prizes associated with the stem cell? What are induced pluripotent stem cells? There are multiple other topics very important from the both prelims and mains exam point of view. Yes. Every cell is specific to that individual. Is it clear? When there is transplantation, you have to match the blood group of the person. Is it clear? Directly you cannot pick up any person to donate blood or bone marrow transplantation. There should be logic of what? The blood group of the individual. Stem cell transplant, bone marrow transplantation means what? What is the purpose of that? Stem cell transplantation. Is it clear? Bone marrow transplantation means stem cell transplantation, where the person's bone marrow is not producing the new blood cells. You are taking the help of some healthy donor and taking the stem cell and transplanting into the recipient. But it is a highly invasive procedure. If you are taking the blood stem cell from person's body, highly invasive, painful procedure, isn't it? Therefore, we avoid that. When it is not possible, we go for those type of transplantation. Yes. See, there are two types of death. One is the clinical death. Is it clear? That is the brain dead person. And another one is the body organs may be functioning. Even after death, heart may be keep on beating. So even after brain dead, we can use the person's transplantation, whether it is the lungs or the liver or the kidney, it can be used for transplantation. Within 24 to 48 hours, we say that is the correct time period for transplantation, provided we are able to preserve those organs before transplantation. So now, having discussed on anemia, we move on to the white blood cells. So we are back to the discussion of immunity. Back to the discussion of what? Immunity. Now, immunity is a direct role played by the white blood cells. So they are the ones who are going to fight against the various infections and diseases. Can WBC fight against cancer? They can fight against infection diseases, we are writing. Can it fight against cancer? How? So they can also kill what? Cancer cells. They can also kill the cancer cells. Now, WBC of different types. For example, the most abundant type are the neutrophils. So about 70% of different types of white blood cell are the neutrophils. Another important type we'll focus are the lymphocytes. So these are highly specialized white blood cells which are very important from exam point of view. So these are specialized white blood cells. So white blood cells are different types, for example neutrophils, for example lymphocytes. There are also other types, but our focus will be on lymphocytes mainly from the exam point of view. The neutrophils, these are involved in we can see phagocytosis. This is about engulfing or swallowing extracellular and kill them. So swallowing, killing and digesting the extracellular microbe, that is called phagocytosis. Here white blood cells there. So for example, this extracellular microbe, example is what? Bacteria, we discussed in the class. Bacteria is what? Extracellular microbe. So to deal with these extracellular microbes, our neutrophils, they will start now, reach the site of infection to swallow the bacteria, kill the bacteria and digest it so that it can be removed from the body. Lymphocytes we will discuss as adaptive immunity. Adaptive immunity. And that is as B cell and the T cell lymphocyte. So lymphocytes are different types. The two important types we'll focus are the B cell lymphocyte and T cell lymphocyte. So till now we have been discussing antibodies, isn't it? It is the B cell that produces antibodies in your blood. So we discussed that antibodies blocking the virus infection or attachment. So these are secreted by the B cell lymphocyte. They secrete the antibodies. To provide protection. Now, before we discuss in more detail B cell and the T cell role, whenever some pathogen enters your body, the first line of defense that starts acting in your body immediately, ready-made immunity, and that is nothing but called innate immunity. So whenever any pathogen enters your body, whether it is a bacteria, it is a virus, then the first line of defense that starts acting in your body immediately, that is the innate immunity. Now, have we discussed the term antigen in the class? Antigen in the class? No. Okay. So this is required to discuss on immunity, because immunity has to recognize, just like security forces, they have to recognize in the country, how do you claim you are the citizen of the country? Someone asks you, how do you claim you are the citizen of India? What will be the answer? First you have to show your voter ID card, followed by your Aadhaar card, passport, isn't it? These are the documents by which you are going to prove. Similarly, immunity means our white blood cells acting against a pathogen. Acting against what? A pathogen, which could be bacteria, which could be virus. Our white blood cell, they should not attack our own kidney cell, liver cell, heart muscle cell. So this means they are able to differentiate that which is a foreign body and which is a self. So WBC should recognize, isn't it? And for recognition, the term antigen plays a vital role in the discussion. So what is an antigen and how it is associated with your immunity? We'll come to the previous slide once again. We'll continue that discussion of the previous slide also. Let's first discuss on the term antigen. Now in the term antigen, where anti stands for antibody and gen stands for generation. Antibody we have discussed, and now you can see relationship with the antigen. Now antigen, it triggers immune response. Antigen is something that can trigger what? Immune response. Or antigen is a substance that can trigger generation of antibody, isn't it? Antigen is a substance that can trigger generation of antibody. This antigen is present in over your body cell surface. You can see here. So surface protein over the cells, that is called what? Antigen. This surface protein or antigen, it acts as a biological marker. It acts as what? Biological marker to provide unique biological identity to a living organism. So it imparts unique biological identity. The antigen that is present over the body cell surface of a living organism, whether plant cell, animal cell or a bacterial cell, it will act as what? Its own self-antigen. So antigen again becomes what? Self-antigen that belongs to the body of that living organism, that belongs to the body cell surface of that living organism is a self-antigen. Any other antigen corresponding to this shall be regarded as foreign antigen or non-self. So for example, bacteria, virus, they act as what? Foreign antigen. Whenever this foreign antigen enter our body, then the body will trigger immune response. Body will trigger what? Immune response. As a result of immune response, suppose some infection enters your body. How does the body respond to that infection? You can find there is redness, there is swelling, there is heat and pain. These are signs of inflammation. So body triggers what? Immune response. And that immune response becomes what? Inflammation is an immune response of your body to protect the body against what? Some foreign antigen. Then gradually, and this is happening, trigger immune response means it is increasing the activity of white blood cells. Is it clear? White blood cells, the activity has increased to protect the body against the foreign antigen. Now this increased activity of white blood cell, they also produce later on the antibodies which will target the foreign pathogen, isn't it? Antibodies, they have to target the bacteria virus to protect the body ultimately. We already discussed that it will block the virus attachment, isn't it? So your antibody mediated immunity starts working. Are able to follow the sequence of event. Some bacteria virus enters your body, it will trigger immune response. Body recognizes as non-self. Recognizes this bacteria virus as what? Non-self. And then will start acting against them by different mechanisms. One mechanism is inflammation. Inflammation will increase the body temperature, make it difficult for them to multiply in your body. Isn't it? Fever is what? A part of inflammation. By increasing the internal body temperature, it is making difficult for bacteria virus to multiply in your body.
So please explain this last part. Antibody mediated.
See, this last part we have discussed, if you remember, that suppose there is SARS-CoV-2 virus that enters your body, and this was the spike protein. What we discussed in the last lecture, that these white blood cells produce what? Anti-SARS-CoV-2 antibody to block the virus attachment or infection and protecting your body. This is antibody mediated immunity, which will block the virus attachment or infection. This is the antibody mediated immunity. Is it clear? That we have already discussed in the first lecture itself. So whenever some foreign virus bacteria enters your body, body has to activate its immune response to protect the body from such foreign antigens. So bacteria virus acting as what? Foreign antigen in your body, where body recognizes non-self. There must be some mechanism how the body will process that antigen, will act against. First we are trying to discuss the term antigen. So antigen is what? It is a surface protein acting as biological marker over the living organism. It will differ from one living organism to another. Plant cell will have different antigen, animal cell will have different antigen, bacterial cell will have different antigen. Now within species also, there are different types of bacteria, different types of virus, where body has to recognize what we discussed. Antibody blocking what? SARS-CoV-2 virus. This antibody will not block HIV virus, will not block dengue virus, will not block Nipah virus. What is the reason? The same antibody which is blocking coronavirus infection will not block Nipah virus. What is the reason behind this? Because antigen of Nipah virus is different. So antibody required will be different now to block the antigen of Nipah virus. So body should produce, immunity should produce diversity of antibodies based on the corresponding antigen. Is it clear? If there are thousands of different varieties of antigen, body also has to produce 1,000 different types of what? Antibodies also to protect itself. Now, our white blood cell will not produce antibody against self-antigen. Our white blood cell, they will not start attacking what? Our liver cell and kidney cells. If it happens, we call it as what? Autoimmune reaction or autoimmunity. If our own white blood cell start producing antibody and harming our own healthy body cells, then it is a breach of the task, and that is called as what? Autoimmunity, autoimmune reactions. So there must be some mechanism that balances our immunity, regulates our immune response. So there are checks and balances for the immunity also, that it should target only what? The foreign antigen, not target the self antigen. If it happens, then it results in autoimmune reactions in your body. That becomes counterproductive against your own body, called autoimmune diseases, autoimmune diseases. So our immunity is able to maintain a balance when it requires to overcome the pathogen, when it requires to remain in silent phase, when it should activate itself. All these are well regulated internally in your body. That we are going to discuss in the role of what? White blood cells. So write down first the term antigen. What is an antigen? Yes. Now, the reason that why we need to regulate fever, even if it is good for the body, the reason is that the activity of these security forces, defense forces in the body must be regulated. If very high fever occurs beyond 100°F to 102, 103, it can damage the brain of the individual. It can be counterproductive again. So, inflammation is good for the body, hyperinflammation is not good for the body. You are asking about hyperinflammation. It goes beyond the limit that body can tolerate. So without causing self-harm, it should try to manage the clearance of infection from the body. So hyperinflammation, that can result in excessive fever. So we have to regulate, body has to regulate that. That is why we have to take that paracetamol to control the fever. Now medicine, see, medicine is paracetamol also we discussed. Medicine is antibiotic also. Both are different medicines. Is it clear? So answer will be different. Paracetamol is not an antimicrobial medicine. It is just to control symptom. Control that inflammation of pain and the fever. Antibiotic is used to kill the bacteria. Now, one prime reason, as I told you, that excessive body temperature, so that can result in means a type of sepsis in the body, where body immune system is overreacting against an infection. So the heart rate will rise to a higher extent, respiratory rate will rise. It can cause respiratory failure and cardiac failure.
For the first time, body instead of body, the antibody will be present automatically, or the body will need to create antibodies first. Then they will.
First, antibody should be produced. Suppose coronavirus was a new virus for us. Had there been antibodies, nobody would have been hospitalized. Is it clear? So for this, what do you need? Vaccine. And what does the vaccine contain? The same virus against which you need protection. Isn't it? Vaccine contains the same virus against you need what? Protection.
There are multiple diseases we know. So we take all the vaccines for that. Is it possible?
Why for every disease we will take the vaccine? For a disease which is life-threatening, that can increase infant mortality rate in the country, maternal mortality rate in the country. There we will develop the vaccine. Is it clear? So write down that antigen is the surface protein. Antigen is a surface protein. Antigen is a surface protein present over the body cells present over the body cell surface of an organism, present over the body cell surface of a living organism. Acting as a biological marker to provide unique biological identity, acting as a biological marker to provide unique biological identity. Known as self-antigen. while antigen present over the body cell surface, antigen present over the body cell surface of other living organisms, including bacteria, virus, and even other human antigen shall be regarded as foreign antigen. Change the para and write antigen may trigger an immune response. Antigen may trigger an immune response. Antigen may trigger an immune response. For example, when a foreign antigen enters our body, it can trigger an immune response. In the form of activation of white blood cells. Activation of white blood cells that will produce or secrete defensive proteins called antibodies. And these antibodies circulate in the blood. To prevent the infection of the virus or neutralize the toxin of the bacteria So this is the concept of self antigen and the foreign antigen. So antigen that belongs to your own body shall be regarded as self antigen. Antigen that belongs to your even parents, brother, sister, outside the family, all will be regarded as what? Foreign antigen. Now as we derive how these antigens are produced, these are surface protein. Everything in your body is produced using the master plan called, what is the master plan of the body? DNA. So DNA has the complete data set, genetic code to make diverse proteins in your body. DNA has that genetic code or biological data, the comprehensive data master plan. And this DNA is derived from where? Both father and mother, isn't it? So therefore there will be some similarity in your DNA in your antigen with your parents, with your brothers and sisters also, derived from the same parents. So there could be similarity in antigen. Similarity in what? Antigen. So that is the reason why during organ transplantation like kidney transplantation, liver transplantation, as we try to discuss today, if you have to select a donor, that donor should be biologically compatible with similar antigen to prevent what? Immunorejection, for better acceptability, compatibility in your body. So that is why whenever the transplantation occasion occurs, then the doctor prescribes what? To look for your own family member, which are biologically related to each other. Not possible in extreme case you can go beyond the family member. Because of what? To avoid the risk of immunorejection. Because higher the degree of what? Foreignness of antigen, greater the what? Rejection. Lower tolerance. So tolerance will be more for similar antigen compared to what? Altogether different antigen. So that is why even in organ transplantation also we apply the same logic, that we should select a donor who is biologically related to us, better from the same nuclear family, of not possible beyond the family member, but extended family members, based on informed consent also. But there have been occasions that when you do not get any donor, then what to do in that case? So there are set SOP guidelines that if they do not get your own family donor, then what you can do in that case. So need for transplantation and who can be the eligible donor. So that is the concept of the antigen. So this concept is important for further discussion on immunity. So this means our white blood cells are able to identify, isn't it, that which is a foreign antigen and can differentiate from a self antigen. They are able to differentiate, they are able to know that a foreign antigen entered the body, isn't it? And they are different from what? Your self antigen. So coming back to immunity, we started with the term what? Innate immunity. What exactly is innate immunity? Now this is very important from exam point of view. Questions are asked from this. This is the most conceptual part, that how our natural immunity works to provide protection. So immunity we have classified as innate or inborn immunity, and this next one is what? Adaptive or acquired immunity. Innate immunity we discussed by taking the example of phagocytosis by neutrophils, where cell was eating the bacteria, removing the extracellular microbe from the body. So whenever some bacteria, virus or any type of pathogen enters your body, the first cellular defense that gets activated to act immediately against that pathogen is called innate immunity. This immunity is present since birth in your body. Since birth it is activated to provide you protection. Therefore it comes under inborn immunity also. What we discussed just now, whenever some foreign antigen enters your body, it triggers immune response, isn't it? Where you have written what? Inflammation. So whenever some injury, infection occurs in your body, whenever some infection or injury that occurs to your body, then the body will show signs of redness, swelling, heat and pain. And this we have written as what? Inflammation. And this is actually the implementation of innate immunity. So this is the implementation of what? Innate immunity. Your innate immunity has started acting now in the form of inflammation. So what will happen? The blood circulation will increase to the area of infection, isn't it? Blood circulation, blood flow will increase in that area of infection, so that more and more white blood cells reach there to fight against infection, clear the infection from there. So innate immunity has started acting. So whether it is a bacteria, whether it is some virus, isn't it? It will give the same type of response. It is giving same type of response. It is not using any such that specialized memory against the antigen. So irrespective of the foreign antigen, it will trigger the same type of immune response. This means it is not using that any specific memory based on the antigen. For example, in case of antibodies, we discussed that antibody produced by the lymphocytes, they are acting against the specific virus antigen. Same antibody is not blocking all the virus antigen. So this means there is some element of memory which is working in the other form of immunity. Here the memory is not working. It is giving same type of immune response to protect the body. So there is no specific memory involved here against the foreign antigen. It is giving me equal protection against the different types of pathogens. Innate immunity, where we discuss like phagocytosis. So whether it is one bacteria or other bacteria, it will try to what? Ingest that bacteria, kill the bacteria and remove from the body in the process of phagocytosis. Now this innate immunity Other examples. The most important example is your epithelial barriers in your body. Epithelial barriers. So for example, through your intact skin, any bacteria virus cannot gain entry. It has to be broken skin. Then only any bacteria virus can gain entry. So whenever there is breach in the intact skin or intact epithelial barrier, there are many important examples of epithelial barrier. One is example your skin is an epithelial barrier. Even your gut is also an epithelial barrier. So unless and until there is some damage here in that barrier, then only the infection can enter the body. So gut, you already know, we discussed like the intestinal epithelium. So these are called as what? Epithelial barriers, barrier organ, which are protecting you, not allowing the infection to enter the body. Then some other form of innate immunity, example, your tears also. So what we are saying, since birth they are acting to provide you protection. Is it clear? As a part of innate immunity. So tears, they contain antibacterial substance called lysozyme. So whether an infant newly born or even adult, tears contain what? Antibacterial substance called lysozyme, which can kill the bacteria. So this is another example of what? Innate immunity. Lysozyme. So lysozyme, antibacterial substance, that is giving the innate immunity here. Already we discussed on the phagocytosis by neutrophils. So immediately after birth, any infection that enters, you can see phagocytic activity inside that newborn body, and also in adult, it is carried forward further. So immunity that is able to manifest itself, implement itself, protect the body since birth, that immunity comes under what? Innate immunity. So all these are present since birth in your body to provide you protection, acting as what? First line of defense in your body. However, with all these acting together, they cannot provide me protection. They cannot provide me comprehensive protection. If our body requires comprehensive protection, means higher level of protection, well-trained level of protection, then we need the help of what? Adaptive or acquired immunity, which involves the role of what? The lymphocytes. So specialized white blood cells acting as what? Memory cells. What are these memory cells? Antigen specific lymphocyte. So what are memory cells? These are antigen specific lymphocyte. Is it clear? So they are antigen specific. That is the reason why we discussed that the memory cell after vaccination of COVID-19, antibodies do not protect me against other viruses. Now, these lymphocytes again of two types. One is B cell lymphocyte, another is what? T cell. T cells again of three types: helper T cell, TH here, cytotoxic TC, and the regulatory or suppressor T cell, T regulatory cells. Let's see the role of T helper cells. Now, helper T cell, they are secreting what? Inflammatory molecules called cytokines. And cytokines they activate B cell lymphocyte and also the cytotoxic T cell. So what is helper T cell is doing? It is secreting inflammatory molecules called cytokines. So these are chemical signals. These are what? Chemical signals which will keep your immunity level higher, to remain protected, to rise to the threshold level, to fight against infection. So these signals, just like whenever a country is attacked by some enemy missile, radar signals, they catch those incoming missile and they activate. So some surface to air missile coming towards India, then India's air defense system has to act, isn't it? By catching those signals, it will activate India's air defense system now, so that it can destroy in the air itself. It can intercept that missile and can destroy in the air itself, isn't it? Just like the Israel protected itself using the air defense system. And there are multiple layers in the air defense system of Israel. What are the layers of Israel air defense system? Everyone knows Iron Dome, isn't it? But even before that, the arrow missiles, they go and intercept to destroy in the air itself, long range. So long range missile attack fired by arrow, medium range fired by David sling, isn't it? And the third one is the Patriot that you have read again in the news. And the fourth one is the Iron Dome. These are the four layers of air defense system of Israel. Similarly, human body also has multiple layers of defense system to protect itself. So when the virus has entered the body and is trying to multiply in the body, then cytokines released by the helper T cell, they activate B cell. So helper T cell acting as a radar signal. Is it clear? Cytokines are radar signals. Then B cell lymphocyte informed to get ready to produce what? Antibodies. To prevent the virus attachment or infection. So B cells are acting to secrete antibodies to block the virus attachment infection, but that is under the command and control of what? Cytokine released by the helper T cell lymphocyte. So they are not acting in isolation, they are acting in coordination. So they are coordinating the role based on what? Helper T cell. Helper T cell is coordinating the action of what? Antibody production based on the infection, followed by inflammation. Now, this was in the context of when the virus is outside the cell. Suppose virus has entered the cell then antibodies cannot protect us, isn't it? Till the time virus is outside the cell, antibody can block the attachment, isn't it? Now we are saying that the virus may have entered the cell. So now we are showing viral replication. So till now we discussed that antibodies, they block the virus attachment. Suppose there is immune escape. There is what? Immune escape. Immune escape means virus has escaped the attack of antibodies. Virus evaded attack of antibodies. And then it becomes what? Virus infected host cell. How does the body protect itself? Have we understood the context? So now we are saying that unfortunately virus that was able to evade the attack of antibodies and it attached to the healthy cell, converted into virus infected host cell. This virus infected host cell becomes biological factory to produce what? So what will happen now? So viral replication, there will be increased viral replication, which will increase the viral load. Causing virulence or severity. So virus was able to escape the attack of antibodies, you have written. Now virus is multiplying in the infected host cell. And that is resulting in what? Increased virus multiplication. Concentration of virus is increasing in the infected body. And that will cause what? Severity of disease or virulence, breathlessness, hospitalization, even death. We have seen that even in many vaccinated people, after giving COVID-19 vaccine, they become infected. But that infection could not translate into what? Severity, hospitalization, deaths. So the question is, how this can be prevented? So first of all, write the term breakthrough infection. Breakthrough infection. What is breakthrough infection? Note down. When fully vaccinated individuals become infected, It is called breakthrough infection. It is called breakthrough infection. When fully vaccinated individuals become infected, it is called breakthrough infection. This may happen due to, write down, mutation or genetic changes in the virus or pathogen. This may happen due to genetic changes or mutation in the virus or pathogen. Whereby the protein of virus or pathogen, changes resulting in immune escape. However, vaccine still protects us. By preventing severity or virulence, and hospitalization. Severity or virulence and hospitalization. But the question is how? Is it clear? So how does the vaccine still protects us? Write down, vaccine primary role is to prevent disease. Vaccine primary role is to prevent disease and not necessarily prevent infection. Have you got the last sentence? Vaccine major role is what? To prevent virulence. Virulence means severity of disease. That people felt breathlessness, getting hospitalized, demanding for oxygen therapy, and many people lost their lives. But after vaccination, when people become infected, they were not hospitalized. They were still protected. Which means they may be infected because of what? Breakthrough infection. Changes in the protein of the virus. When the virus multiplies, it will change its protein. Suppose coronavirus multiply, it changes its what? Spike protein. If spike protein slightly changes also, then the antibody not able to block the virus attachment. And virus able to escape the attack of antibodies called breakthrough infection, immune escape. That is what immune escape. Still our immunity, vaccine induced immunity is still protecting us. But how that protection comes to the body? That comes because of the role of cytotoxic T cells. They kill the virus infected host cell. Along with that, they also break the virus into fragments. They will destroy this virus infected cell, and they also break the virus into fragments. And this is known as, so it is the role of cytotoxic T cell. This is called cell-mediated immunity. This is called what? Cell-mediated immunity. Where the T cells, cytotoxic T cells, they kill the virus infected cells and they also have the ability to kill the cancer cells. This is a very important point. They not only kill the virus infected cell, but they have also the ability to kill the cancer cells in your body. These two are very important role that you must remember, that it is the cytotoxic T cell or cell mediated immunity that is preventing that severity even after breakthrough infection. So what we discussed, breakthrough infection, what is happening breakthrough infection? Virus is evading attack of antibodies, enters the cells, multiplies in the cell. However, still our immunity is protecting us, and that is protecting through what? The role of cell mediated immunity. It is the cell mediated immunity or T cells which will kill this virus infected cell, will not allow the free multiplication of the virus, will not allow the free flow of the virus in your body. It will destroy this infected cell. So without cell, virus cannot multiply. It will also break the virus into fragments, ultimately to help clear the infection from your body. Keeps a check on your infection now. Does not allow the infection to get translated into severe disease or virulence. So answer is what? Cell-mediated immunity. We'll continue the next class from cell-mediated immunity. How does these T cells, they kill this virus infected cell? That mechanism we'll follow in the next class.